Are introducing brainstem and also getting into the lowest part of the brainstem called medulla oblongata. Our reading material comes from Siegel and Sapro, Essential Neuroscience, Chapter 9, Brainstem 1, the medulla. As you can see, I'm giving you 5 learning objectives. The first 2 refer to general organizing features of brainstem, and then the last 3 delve into medulla oblongata. As far as organizational features of brainstem are concerned, we use the number 3 as a really useful number. And that is for many reasons, such as the anatomical and functional subdivisions of the brainstem, starting with the fact that we have 3 components: medulla, pons, and midbrain anatomically. And functionally, we can also distinguish between cranial nerve functions, integrative functions, and conduit functions of the brainstem. We also need to understand that these functions overlap. Cranial nerve functions and cranial nerve nuclei are organized at various levels of the brainstem, and they have sensory as well as motor functions. Integrative functions refer to something that may link sensory to motor and even some higher order mechanisms, such as regulation of pain and regulation of consciousness, but also cardiovascular and respiratory functions would be included in this. Conduit functions make all of these possible. Think all of the descending and ascending projections. Motor and sensory projections may be some of our long tracts, maybe some other tracts that either originate or terminate at the brainstem, connecting spinal cord to brain, brain to spinal cord, and various portions of brainstem itself. Think of the wiring system organized by way of these conduit functions. Now, components of ventricular system can be observed in various parts of the brainstem, such as up in midbrain, a very cylindrical cerebral aqueduct. The fourth ventricle opens up when we are in the pons, and the upper or rostral part, also known as open part of medulla oblongata, and further caudally in the closed portion of medulla oblongata, we will see the central canal, which then also continues caudally into the spinal cord. Again, using our favorite number, the number 3, there are 3 pairs of cerebellar peduncles connecting the brainstem to cerebellum: the superior, the middle, and the inferior cerebellar peduncles. We will see these later on. Looking at the internal organization of brainstem, we can again use our number 3 to say from dorsal to ventral, tectum, tegmentum, which is in the middle, and the base or ventral aspect of brainstem, keeping in mind that tectum is really only observed in midbrain, so the uppermost portion of brainstem. Important facts about brainstem. Of course, brainstem is rostral to spinal cord as we go further rostrally all the way to midbrain, and it becomes caudal to diencephalon. So diencephalon, maybe the two halves of thalamus would be represented here in brown, and just caudal to it, we get into this brainstem, the stalk-like elongated portion of the brain going toward spinal cord. So between diencephalon and spinal cord, we will find the brainstem and the various portions of ventricular system in the brainstem. The central canal, this little dot that we can see here, and then the wide open fourth ventricle in rostral medulla and pons, and then cerebral aqueduct as we go into midbrain. Now the floor of the fourth ventricle here is called rhomboid fossa. That just came in as an annotation in blue to signify that the floor of rhomboid fossa, the floor of the fourth ventricle, the rhomboid fossa. Next is the idea that the brainstem is connected to the cerebellum, once again, by the three pairs of cerebellar peduncles. These are illustrated here as the cerebellum has been removed. The cerebellum is still attached in this illustration, coming from the ventral point of view. Here, from the dorsal point of view, the cerebellum has been removed, leaving the cut profile of the SCP, or superior cerebellar peduncle, the MCP, very large, middle cerebellar peduncle, and then the ICP, the inferior cerebellar peduncle, is really coming in from medulla oblongata. We see a cross-section over here, a very prominent lateral bulge, the inferior cerebellar peduncle from medulla. So once again, the three pairs of cerebellar peduncles. The tegmentum, the inner core of brainstem, contains cranial nerve nuclei. In fact, from oculomotor number 3 to hypoglossal number 12, midbrain, pons, medulla, eventually along the rostrocaudal axis, we'll find these cranial nerve roots attached to brainstem, keeping in mind that the first two, olfactory and optic, are attached to forebrain. So we will only go from 3 to 12 as we travel from midbrain to medulla oblongata, if you will, as if we were climbing rungs of a ladder, going up or perhaps descending. Now, very importantly, the lower portions of brainstem, particularly pons and medulla, contain life-sustaining respiratory and cardiovascular centers. So that is why we need to pay very close attention to the normal as well as abnormal functioning of these regions of the brain. Going to anatomical features of brainstem. Externally, we can begin in an isolated brainstem from the ventral view and observe the anatomical features. Or we can also look at the brainstem dorsally, again with the cerebellum removed, so we are seeing cut profiles of some of our cerebellar peduncles, but a wide open view to the rhomboid fossa. And then the rest of the brainstem, we can also observe it from a mid-sagittal view. Now you can also notice that these blue lines, which are perpendicular to the long axis, signify various rostrocaudal levels. At these levels, we can make cross sections and look inside. And we will in fact do this. We will have to do this to interpret internal structures, nuclei as well as pathways. We will have to interpret them. Now, a quick study of dorsal and ventral views of a human brainstem is in fact contained in the YouTube channel, the WCU.com Neuroscience YouTube channel, so I recommend you to look at that. This is a portion about the cross sections, the internal organization of brainstem. You could go from caudal to rostral. So from caudal to rostral we can go. Also we can go from caudal to rostral. That would correspond to a typical level of medulla. Up here, maybe a mid-medulla in this case. Sort of a caudal mid-level pons has been selected, perhaps from about here, and then higher up in the midbrain, about here from superior colliculus levels, another typical level of midbrain cross-sectional brainstem can be observed, and this is to say that we have tectum, tegmentum, and base. Tectum, as I said earlier, is only visible in midbrain. This is in fact the tectum dorsally, superior and inferior colliculi of midbrain, and a very broad central region throughout. We call that the tegmentum. It is a central region relative to the surface of the ventricular system. That would be here, and once again here, relative to that, a broader central area, and then ventrally the base or ventral aspect throughout the brainstem, midbrain, pons, medulla oblongata. Also noticing that the proportions change all the time. The base, the ventral aspect, is very, very prominent in the pons. At least 2/3 of the pons, and I am pointing here, once again, I'm pointing here, at least 2/3 of the pons is ventral pons, corresponding to the base. That is because of pathways and some unique structures that occur in the pons. So all of these proportions will change, and it is a good idea, generally speaking, to follow the ventricular system and the shape of sections to understand the level we are looking at. So with that, again, understanding that both the cranial nerve nuclei and reticular formation are embedded in the tegmentum. Here's an example of reticular formation. These are various groups, conglomerates of neurons, some of them chemically coded, containing monoamines and other neurotransmitters, some of them with very long and extensive projections going up and down. That is your reticular formation fulfilling many integrative functions in brainstem, and then more discretely dispersed will be congregations of cranial nerve nuclei. Long pathways, and those are illustrated under the conduit functions, are usually but not always close to the ventral aspect, the base. And again, some of them ascending, some of them descending. Think ascending sensory pathways, spinothalamic, dorsal column, medial lemniscus, or descending ones, such as corticospinal tract. But it could be rubrospinal or reticulospinal, and all the rest. Typically, we focus on corticospinal, and that is very close to the base indeed. We will also be watching for unique structures that appear at certain levels of brainstem. For example, we may find the inferior olive in the medulla, some other features of pons, parabrachial nuclei, maybe other conglomerates of neurons, such as locus coeruleus, that will be in the pons, substantia nigra, periaqueductal gray, when we are up here in midbrain. They are unique features that come and go. They appear and disappear from our sectioning plane as we make these cuts. We will be making these cuts later on. And even here, there is an example of these various levels of brainstem for the conduit functions of brainstem. So very importantly, we have stable and predictable localizations of pathways. These are very important for localizing effects of lesions. They are very predictable and very repeatable in all humans. For example, when the corticospinal tract is damaged, that would be depicted in red in this illustration. So our reticular formation, of course, is not a pathway. It is just for reference, showing you where most of the tegmentum is. So you focus on that purple, whether you're in the medulla, so A and B levels are medulla, whether you're in the pons for C and D, and E and F for midbrain, the central portion contains the tegmentum. And relative to that, we have a fairly stable localization of pathways that will go up and down. It is very critical that we all must have an understanding of this early on. And then we'll go a little deeper. So starting with this very large descending tract, the corticospinal tract or pyramidal tract, when you're in the medulla, you can see that in the medullary pyramid, but also in the pons, also in the midbrain. This pathway is very close to the ventral aspect. Now we also have the spinothalamic tract, more laterally organized, anterolateral system, as it's often referred to. Once again, it, in a way, relative to the tegmentum, retains its anterolateral position. Even when you're in the pons, even when you're in the midbrain, there is just a much larger base. So once again, going back to an earlier point, that proportions will change. And some of the organizational features are relatively stable as far as the pathways going through. And the posterior columns, which become medial lemniscus after that first synapse, so we trace the medial lemniscus, they also occupy this central position depicted in blue. So some of our most important descending or ascending pathways. Of course, deficit of these pathways will be translatable into very distinct motor and sensory deficit signs. Lesions of these pathways, in other words. What is the reticular formation? I've already made some references to this, that this is an integral part of the tegmentum along with cranial nerve nuclei. So these are cell groups presenting in the tegmentum. There is typically a more rostral component here. Blumenfeld labels that in purple, and a very extended caudal reticular formation that even goes into the spinal cord in the intermediate gray. As I mentioned before, these are implicated in integrated functions of central nervous system, such as regulation of consciousness, sleep, wakefulness, regulation of pain, but they also regulate reflex activity, muscle tone, extensor tone, things of that nature. Think of reticulospinal tracts that originate from pontine and medullary levels that we already mentioned under spinal cord. That type of thing, regulating reflexes and extensor tone, but so many other functions. So many other functions are also concentrated here, especially looking at our chemically coded neurons, such as the monoamine containing cell groups, serotonin, norepinephrine, dopamine would come to mind. Some acetylcholine cell groups will come to mind that live in cell groups within the reticular formation, and they have either ascending or descending projections. Some of these projections can be very diffuse, and we cannot see them individually as distinct pathways. They can only be identified by specialized methods, looking at those neurotransmitters. And of course, pharmacologically, they can be approached and observed. At this point, having studied the general organizing features of brainstem, we can delve into medulla oblongata. Of course, medulla oblongata being the lowermost part of a brainstem, coming from the perspective of spinal cord, just leaving the edges of foramen magnum and transitioning into the cranial cavity. In fact, the spinal cord ends there. Medulla will begin there, and it will go all the way to the ventrally very nicely observable pontomedullary sulcus. So this is once again a ventral view, and we are observing the boundary between the medulla and the pons as pontomedullary sulcus. There are several features on the ventral side that we need to see. For example, cranial nerve roots from glossopharyngeal number 9 to hypoglossal number 12 are in fact attached to medulla oblongata. These are also known as cranial nerves of medulla oblongata. It is a good idea, even clinically speaking, to group them because lesions may occur together as a lot of the cell groups giving rise to these nerves live inside the medulla oblongata. So we call them cranial nerves of medulla oblongata. There will be a separate designation for nerves lined up at the pontomedullary sulcus, these nerves. More about that later. So once again, just focusing on medulla from here to here, or from here to here, caudal to rostral, or rostral to caudal. In this ventral aspect of medulla, we can see the anterior median fissure. We have seen that in the spinal cord as well. We retain it even as we go into the medulla. These two bulges, the pyramids, the medullary pyramids, the pyramidal decussation that may be subtle in an anatomical, actual cadaveric specimen, very subtle. The decussation near the border of spinal cord and medulla, very caudal medulla, the decussation of the pyramidal coracospinal tracts. As I'm hovering over here, the olive of the medulla is a bulge, kind of ventrolaterally, if you will, leaving the pre-olivary and post-olivary sulci, where nerve rootlets are attached. So the hypoglossal nerve is in fact attached to the pre-olivary sulcus, and the glossopharyngeal and vagus nerve roots are attached to post-olivary sulcus. It's a little bit um, out of our view because we would have to rotate the brain. We can do that in the laboratory, or you can also watch some of my videos on the YouTube channel for this. So once again, ventral features, not too many, but they are quite important. Here's medulla oblongata from the dorsal point of view, again with the cerebellum removed. Midbrain pons, we are focusing on medulla, noticing the open medulla as opposed to the caudal medulla, RM, or rostral medulla, or CM, also known as caudal medulla, with some imagination reminding us of a scoop. The open aspect of that scoop would overlie the caudal part of rhomboid fossa, the floor of the fourth ventricle. That's the open or rostral medulla. And then the handle, if you will, of our scoop is the caudal medulla. It becomes closed in the sense that it contains the central canal as the ventricular system is continuing. And of course, central canal is also found in the spinal cord, further caudally. So we observe this to make a distinction between the open or rostral part of the medulla and the closed or caudal part of the medulla. Once again, looking at our dorsal view in an isolated brainstem, we start our medulla here from the lateral recesses of the rhomboid fossa. That would be over here. One on each side because, in fact, laterally, this will translate into the foramina of Luschka, the lateral openings of the fourth ventricle, as the fourth ventricle is communicating with the subarachnoid space. So that anatomically corresponds to the lateral recesses of the rhomboid fossa, and hence the fourth ventricle itself. And from this point on, and caudally, we start our investigation in medulla, starting with the open medulla, and then at the level of the obex, which is the caudalmost point in the rhomboid fossa, we continue caudally, and we are, in fact, in the caudal or closed medulla. We are, in fact, somewhere here. Now looking at the ventral view. So it is very critical that you recognize this dorsally from a specimen with the brainstem shown here with the cerebellum removed, giving you full access to the rhomboid fossa. Okay, with that in mind, we can start caudally here in the medulla, focusing on the obex, the caudalmost point of this lower triangle overlying the open medulla. That's your obex. And we look around, we see that the dorsal columns, the fasciculus gracilis and the fasciculus cuneatus, the dorsal columns carrying conscious proprioception, fine touch, vibratory sense from spinal cord that we observed, they continue ascending and they form these tubercles, these bumps, where they terminate, and these tubercles contain the nuclei for the dorsal columns. So those are the gracile and the cunate tubercles. Now, once again, the rhomboid fossa is the floor of the fourth ventricle. We see area postrema, a chemosensitive area, just surrounding the obex, and two triangles, the vagal trigone and the hypoglossal trigone, just adjacent and still medial to sulcus limitans, containing the bumps corresponding to cranial nerve nuclei, signifying vagus and hypoglossal motor nuclei. Of course, you might know this already from our study of embryology, but we will get to it again in the study of cranial nerve nuclei. The hypoglossal is a somatic motor, and the vagal nucleus will be an autonomic or visceral motor nucleus. We will look at these features also in an internal view. So we'll watch for that. And also keep in mind that you can review this using the YouTube channel for WCU.com Neuroscience. Now, eventually, we need to get to this more integrated, semi-transparent view of the brainstem, and in it, the medulla oblongata, by imagining pathways running through, and also imagining cranial nerve nuclei, some of the motor ones and many of the sensory ones outlined here. Again, this is medulla from here to here. Noting the ascending and descending pathways. Ascending pathways coming up. Dorsal column, medial lemniscus pathway. For example, corticospinal tract is coming down, crossing the midline at the pyramidal decussation here. So those are some of our ascending and descending pathways, and our intrinsic components would be the cranial nerve nuclei that live in medulla oblongata, keeping in mind that for the most part associated with hypoglossal, vagus nerves, glossopharyngeal, and in upper medulla, the beginning of cranial nerve 8, vestibulocochlear nerve associated nuclei. Trigeminal is very unique. We'll get to it in its own right. It has its own intricacies, so just note that trigeminal exists. We don't have to delve into it very deep, but we will be. So there will be several of these cranial nerve nuclei serving sensory functions. A really big one is trigeminal and some of our special sensory. So with that in mind, a very useful quote from Blumenfeld saying, "There is no substitute when learning brainstem anatomy for examining its detailed structures in stained sections, serial sections of the human brainstem, We will refer to these sections as we review different nuclei, pathways, and functional systems in this chapter, says Blumenfeld in his own chapter on the brainstem. For the next series of slides, we need to recognize and differentiate levels of medulla oblongata from cross sections. We should be able to localize main groups of cranial nerve nuclei. As I said before, we would be anticipating things like hypoglossal and nuclei belonging to vagus and glossopharyngeal nerves for the most part. We also need to keep in mind the most important long pathways, long tracts, such as corticospinal, such as dorsal column and dorsal column nuclei, and then medial lemniscus from the tubercles on, and rostral to those, from those nuclei. It becomes medial lemniscus pathway. And we also keep in mind that we have the spinothalamic tract already crossed and ascending from spinal cord. Of course, spinothalamic tract carries nociception and thermal information. I do have a very quick, unedited video on the YouTube channel called Coronal Slicing a Brainstem, just showing you various levels of midbrain pons and medulla, the way they look in an unstained cadaveric brain specimen. So I recommend that. This would be a good way of learning the shapes and the proportions of brainstem. In this illustration, under A and B, we are looking at the caudal or closed medulla. First, very caudally at the spinal medullary junction, where the decussation of the pyramids is. And the dorsal columns are still coming in, practically uninterrupted from spinal cord. So these are collectively the dorsal columns or posterior columns, specifically fasciculus gracilis from lower limb information, and fasciculus cuneatus bringing information from upper limb. And once again, the very important pyramidal decussation at this level. Just a touch further rostrally, still in the caudal medulla, we do see the nuclei of gracilis and cuneatus corresponding to the tubercles that we observed gross anatomically. So the tubercles contain the nuclei where the second order neurons live, and that is an important second type of decussation now a sensory decussation. Earlier we had a motor decussation here for the pyramid, but a little higher up, the decussation of the dorsal column pathway, from which point on it becomes medial lemniscus as it loops up, and this will go, of course, all the way to the thalamus. So these are two important levels that we can think about and we can observe. Now, as far as navigation, the open medulla, the mid-level medulla, we are making a cut at a level where the fourth ventricle is already open, and we can line up the cranial nerve nuclei, if you will, from medial to lateral. The medial most are motor nuclei. Somatic motor is always medial most, and then the visceral motor. A little deeper, the branchial motor or SVE component, as it's known. And then just lateral to sulcus limitans, we are getting into the sensory components of these nuclei belonging to these various nerves. So all the way to cranial nerve 8 and lateral most, which is the special sensory column, if you will. In this particular case, it's corresponding to vestibular functions, and then at even more rostral levels, auditory functions from cochlear nuclei. But at this point, I would like you to look for a sulcus limitans first. The sulcus limitans corresponding to the division between the medially located motor cell groups, the hypoglossal and vagal motor nuclei, and then the visceromotor, and then the viscerosensory, as well as the other sensory cell groups more laterally, lateral to sulcus limitans. So look for that sulcus limitans to separate motor from sensory. Still at this level, we can look for pathways, myelinated bundles, important long tracts, the corticospinal or pyramidal tract running in the pyramid of the medulla, ventralmost. The medial lemniscus stands out here, and also the spinothalamic tract in that ventrolateral, so-called anterolateral position still. In addition to these long tracts, for the first time ever, I'm also pointing to a new tract for us, the medial longitudinal fasciculus, or MLF, medial longitudinal fasciculus up here, a paired fiber bundle. These will be important in more rostral levels of brainstem for the coordination of eye movements. And then in these lower levels of brainstem in the medulla, some of the vestibulospinal projections go down. So this is also an intrinsic superhighway system for coordinating certain functions. Once again, whenever we think of MLF, or medial longitudinal fasciculus, I would like you to think of either eye movements or some projection going down to spinal cord, regulating extensor tone and orientation of head and neck. When we're going in these pathways into cervical segments, for example, MLF, medial longitudinal fasciculus. Medial longitudinal fasciculus. Our final slide here will be an even more rostral level of the medulla, very close to pons, in fact, very close to the pontocerebellar angle. This portion of the medulla is wide laterally and dorsally because the inferior cerebellar peduncle becomes more prominent. Notice that. The inferior cerebellar peduncle, carrying important pathways, many to cerebellum from spinal cord. For example, our dorsal spinocerebellar pathway coming from the nucleus dorsalis of Clark that we observed in spinal cord, but also other pathways such as some of the vestibular connections. So once again, which is to say that the lateral portion of rostral medulla is very large because of the inferior cerebellar peduncle, and also because of the fact that vestibular and cochlear nuclei belonging to cranial nerve 8, vestibulocochlear nerve, are coming into the sectioning plane. And you may even trace the vestibulocochlear nerve itself. So that would be something to observe. And other long tracts and features such as the olive, the inferior olivary nucleus, medial lemniscus, MLF, are fairly stable still at this level. Just note the laterally extended medulla, dorsally and laterally extended medulla, because of the inferior cerebellar peduncle and the vestibular complex, as well as our cochlear nuclei. And we will do this just before we transition into the pons. So this is the final slide for medulla. Next time we meet, we will be going into the pons. Thank you for your attention, and we will meet again when we're discussing the rest of the brainstem.